Dear students, Assalamu alaikum. Today we will discuss about the genetics of the bacteria and the viruses. Especially our focus will be on the bacteria. The chapter outlines viruses and the bacteria in the genetics of the virus and bacteria, mechanism of genetic exchange in bacteria, and evolutionary significance. Bacteria and viruses in genetics. These are small size organisms up to less than or approximately up to the 2 micrometers. They can reproduce rapidly and they can be reproduced on the selective media. So, for example, the media containing the antibiotics. Relative simple structures and the physiology. They have simple structure and they have because they are the unicellular organisms, so their physiology is to some extent simple. They have complete genome sequences, and then their complete genome sequences are known. Most of the microorganisms is known. The complete genome sequences of the most of the microbes is known. Genetics of virus can only reproduce by infecting living host cells. So, for example, the bacteriophages are the virus that infect the bacteria. Several important genetic concepts have been discovered through the studies of the bacteriophages. Bacteriophage, for example, the T4. This is the double strand. This organism contains double-stranded linear DNA genome and a protein head. It contains its genome contains 168,800 base pairs and 150 characterized genes, and it can go towards the lytic phase. It can go through a lytic cycle because this is a lytic phase. This organism initially, when infect the bacteria, after infecting the bacteria, it injects its DNA into the bacteria. Within the 2 minutes, the synthesis of the phage-specific mRNA is started. In the 6 minutes, replication of the phage DNA begins, and the host DNA has been degraded by the phage endonucleases and encoded nucleases. Phage nucleases degrade the host genome and replicates its self genome. After 14 minutes, DNA filled in the heads and the tail lacking five. Uh, Within the 14 minutes, the proteins are synthesized and DNA is packaged into the head of the virus. The tail lacking tail fiber and assembled tail fiber first appear. In 17 minutes, first intact phage particles are assembled. Intact phage particles are assembled, and within the 25 minutes, the host bacterium is lysed, released about 300 progeny phages, and they are now ready to infect the other cells. From the single cells, this much phage particles are released. Bacteriophage lambda, this is a double-stranded linear DNA genome that it contains. Uh, Genome contains 48,502 base pairs and about 50 genes. It may go towards the lytic and the lysogenic cycle. It may be in the both forms. So, for example, lambda phage, in fact, the E. coli. Lambda chromosome, when inserted into the E. coli, it can go into the lysogenic as well as the lytic cycle. In the lytic pathway, the multiple copies of the genome of the lambda is generated, and after the generation of the multiple copies, these DNAs are packaged into the protein codes. The virus completed after its assembly, and then it go towards the lysogenic cycle. It goes towards the lysis of the cell. While as far as the lysogenic pathway is concerned, the lysogenic pathway, the genome of the virus is incorporated into the genome of the bacteria, but by the site-specific recombination, and this genome of the lambda prophage is incorporated into the genome of the bacteria and remain until the situations are favorable. ये लाइसोजेनिक फेज में चला जाता है जब अपने आप अपना जीनोम जो है वो इनकॉर्पोरेट करा लेता है इन दी बैक्टीरियल जीनोम। तो जितना बैक्टीरिया डिवाइड करता जाएगा इतनी ही जो कॉपीज हैं इसकी जीनोम की डिवाइड होती रहेंगी, लेकिन ये अभी तक अपने प्रोटीन कोड्स और अपने अपने पार्टिकल्स नहीं बनाता। पार्टिकल उस वक्त बनाएगा जब बैक्टीरियल सेल जो है वो अनफेवरेबल कंडीशन में चला जाता है, उस वक्त ये अपनी पार्टिकल्स को बनाता है और अपने आप को असम्बल करता है उस पार्टिकल में, और फिर लेटिक फेज में जाकर तो ये जो है वो वहाँ से सेल्स को बर्स्ट करके निकल आते हैं। The combination process, this is the size, it is actually the site specific process. There are specific sites on the genomes, both on the virus and the bacterial genome, where the combination can take place. These sites are known as the ATTP, P for the phage virus, and ATTB, B for the bacteria. It means ATT sites on the phage and ATT sites on the bacteria. Lambda integrase enzymes is actually required for this integration. Linear package form of the lambda chromosome showing the location of the few genes here. This is the ATTP site and this is the ATTB site. The combination between these two ATT sites takes place and the viral genome is incorporated into the bacterial genome. Genetics of the bacteria. Bacteria contain genes that mutate to produce altered phenotype. Genes transferred in the bacteria is unidirectional from the donor to the recipient cells, monoploid to monoploid. Bacteria contains one main chromosomes with a few thousand genes, variable number of plasmid and episomes, asexual reproduction by simple fission, and the parasexual transformation of the DNA. Phenotypes in the bacteria. Bacteria mostly forms the colony. Colony is equal to can be considered as the one bacteria. Colony color and the morphology is dependent upon the define the characteristics of the bacteria, and the nutritional mutations nutritional mutants for energy sources can be produced in the bacteria. Prototrophs can produce any metabolites, and auxotroph do not produce specific metabolites. We can grow the bacteria on the antibiotic resistant media. Say for example, this is the media, bacterial colonies. This is selective media for the bacterial colonies. The red ones are the Syringia marcensis, and the light bluish colors are the Pieranginosa. Recombination in the bacteria can be take place by the two processes. With the help of the two processes, there can be a double crossover or there can be a single crossover. And if there is a double crossover, say for example, a bacteria is containing the A negative gene, and the second gene, which is the A positive, is um, donated by some other bacteria. The gene transferred in the bacteria produce a partially diploid recipient cells. There is a partial diploidy condition in this bacteria where the genome is inserted. There is a partial diploidy condition. After the two crossover, insert a segment of the donor cell's chromosomes into the intact cells, circular chromosomes of the recipient cell, and replace the recipient fragment. You can say this blue segment, this A negative genes is replaced by the A positive, and the A negative will be degraded. In the single crossover gene transfer produce a partial diploid bacterium, single crossover. Um, A single crossover between the linear donor fragment and the intact circular chromosomes of the recipient produces an unstable linear chromosome. Mechanism of the genetic exchange in the bacteria. Bacteria exchange genetic material through three different sexual processes. You people know these processes are the transformation, conjugation, and the transduction. Bacteria exchange genetic material through three different parasexual processes. Transformation, uptake of the free DNA, A free DNA is taken from the environment. You can see an environment and inserted into the genome of the bacteria. The second process is the conjugation, which is direct transfer of the DNA from one bacterium to another. A donor cell transmits its genome to the recipient cell. The third way is the transduction, transfer of bacterial DNA by bacteriophage. A bacterium, a virus lies a bacteria and carry its genome to the other bacteria. Sometimes the genome of the bacteria can also be transported with this virus. You can say the virus, when packaged into its when packaged its genome into the viral head, sometimes it takes the genome of the donor bacteria, and when it infect the other bacteria, it will transfer that genome into the new bacteria. The YouTube experiment. In the YouTube experiment, a U-shaped tube is formed. A U-shaped tube. On one side, a pressure pump is attached, and on the second side, cotton plug is attached. In the inner side, at a point, centered glass filter bacteria can a glass filter is attached. A filter is attached where the bacteria cannot pass through the filter, but virus and the DNA can. So when the pressure is applied to mix small particles, if recombination occur, it means it do not require the cell-to-cell contact. When the pressure was applied, some bacteria become, which were a negative, they become a positive. So it means that direct in contact of the bacteria is not required. Transformation Staphylococcus pneumoniae. The example of the Staphylococcus pneumoniae is very important. Polysaccharides capsule containing bacteria is the S type, and non-polysaccharide capsule containing is the R type. It means the polysaccharide containing bacteria will be smooth and the other will be non-smooth. Other will be the rough type. Okay. In this experiment, the rough form was the virulent, while the smooth form was the virulent form. First type S bacteria which were encapsulated was injected into the live mouse, and the mouse get the disease and was dead. So after some time, another mouse was taken, and heat killed type 3S bacteria was injected into the mouse, and it cannot affect the mouse. Mouse still was alive. The third living type R bacteria was injected into the mouse, but it cannot cause disease because these were the virulent. After that, then both the heat kill type 3S and living type 2R was injected into the mouse, and the mouse was found dead. What was happened behind this? Because the dead bacteria, the components of the dead bacteria was in the solution, and the DNA was transferred from one bacteria to the other bacteria, and convert the other bacteria into from the R type to the S type. Because of this conversion, the R type become the virulent and caused a disease to the mouse, and the mouse was dead. Transformation in the Bacillus subtilis, DNA from donor cells to the DNA from the donor cells to the recipient cells. Transformation in the Bacillus subtilis. For the transformation, a competent bacterium can bind exogenous DNA and transport it into the cell. All the bacteria cannot bind with the external genome. When the exogenous DNA is bounded to the receptor, complex by the competence protein COM-EA and COM-G. This is very important. Complex with the exogenous DNA is bounded to the receptor complex by the competence proteins. These competence proteins are one is the EA and second one is the G. Here you can see the DNA is bounded with the competence protein. Here is the EA and second one is the G. This is present in the cell wall of the bacteria. After this combination, as DNA is pulled through the channel composed of the protein COM-EC, COM-EC, this is the channel which is made by the protein COM-EC. This is the channel which is made by the this is the channel made by the proteins EC. Okay, when once the DNA is entered to the COM-EA and COM-G protein, then it interact with the COM-EC. As the DNA is pulled through the channel composed of the protein EC in the membrane by the COM-FA DNA translocase. While for the passing from this channel, the protein FA is required. While this transport, one strand of the DNA is degraded by the deoxyribonucleases, the surviving strand of the DNA is stabilized by a single-stranded DNA binding protein and REC-A protein, recombination complex. ओके, दोबारा से एग्जांपल को क्लियर कर रहा हूँ जी। जैसे ही डीएनए इसके साथ इंटरेक्ट करता है, इंटरेक्ट करने के साथ ही कुछ प्रोटीन कम्प्लेक्सेस हैं जो इसको ज्वाइन करती हैं, जिनमें कॉम ई ए जी सेल वॉल पे प्रेजेंट हैं, ई सी जो है साइटोप्लाज्मिक मेम्ब्रेन में है और वो उसके साथ इंटरेक्ट करती है कॉम एफ ए। कॉम ई ए प्रोटीन्स के साथ इंटरेक्ट करके डीएनए इन साइड पे मूव होता है और वहाँ पे वो इंटरेक्ट करता है कॉम ई सी चैनल के साथ। कॉम ई सी चैनल से कॉम एफ ए प्रोटीन उसको ड्रैग करके इनसाइड ऑफ द सेल लेकर आती है, लेकिन साथ ही में वहाँ पर डीऑक्सीराइबोन्यूक्लियेजेस प्रेजेंट होते हैं जो एक स्ट्रैंड डीएनए का डिग्रेड करते रहते हैं, एक स्ट्रैंड इनसाइड पे ट्रांसफर होता है। जैसे ही ये एक स्ट्रैंड इनसाइड पे ट्रांसफर होता है, फौरन से ये सिंगल स्ट्रैंडेड डीएनए बाइंडिंग प्रोटीन के साथ बाइंड करता है। सेकेंडली इसके साथ रेक ए प्रोटीन्स बाइंड करती हैं। The single strand of the donor DNA is integrated into the chromosome of the recipient cell, producing a DNA heteroduplex with different alleles in the two strands. ये जो सिंगल स्ट्रैंड इसके अंदर आता है, ये आकर यहाँ पर कम्प्लेक्स, ये आकर यहाँ पर बाइंड कर जाता है इस क्रोमोसोम के साथ, यहाँ पर आकर ये अपना कम्प्लेक्स बनाता है। दिस इज एक्चुअली द हेटेरोडुप्लेक्स. Heteroduplex is a double-stranded molecule of the DNA originated by the recombination of the single complementary strand derived from exogenous sources. Conjugation in the E. coli DNA is transferred from donor to the acceptor cell through specialized intracellular structured conjugation tube. Conjugation tube इसमें बनती है जिसको आप F pili भी कहते हैं. Donor cells have specialized cell surface appendages called the F pili. F pili synthesis is controlled by several genes on the small circular DNA molecule or the mini chromosomes, which is called the F factor. The F factor in the E. coli F factor exists in two different forms. F factor exists in the two different state, F factor autonomous and an integrated state. The autonomous, it can replicate independently in the host chromosome. An integrated state covalently inserted into the host chromosome and replicate with the host chromosome. If it is autonomous, it can replicate autonomously, and if it is integrated F factor, it will be integrated into the chromosome of the host organism, and there it will replicate. Cells containing the F factor in the autonomous state is called the F positive cells. When the F positive and the F conjugate only F negative are the conjugated. When F positive conjugate with the F negative, only autonomous F factor is transferred and both become F positive because F factor replicate during transfer. Formation of the HFR strains, F factor can integrate at any site by site-specific mechanism. Site-specific mechanism, you can say there are some specific site where it can be inserted. The integration of the F factor mediated by the IS elements. IS element, I will describe it in the later chapter in the transposons. Cells having integrated F factors are called the HFR high-frequency recombinations strains. An integrated state F factor mediate transfer of HFR cells chromosomes to the recipient F negative. An integrated state F factor mediate transfer of HFR cells chromosome to the recipient to recipient F negative. Only a portion of chromosome is transferred before cells separate, thus breaking the chromosome. When the cells separate, the breaking of the chromosomes take place. Rarely entire chromosome is transferred. It means when it means when two bacteria conjugated, when the two bacteria come in the interact with each other, when the two bacteria come in interact with each other, the part of the genome which will pass through this recombination tube will join the other chromosome. If this conjugated tube is broken down, the remaining part will not transfer into the bacteria. So, there are very real chances that the complete genome of the bacteria is altered. F factor can integrate at any F factor can integrate at many different sites by the site-specific mechanism. Site of integration determines the origin of transfer, where it need to be attached, where it will be attached. Uh, it is determined by the site origin of the transfer from where it is transferring. Orientation of F integration DCBA reading clockwise or ABCD counterclockwise is determines whether the sequence of chromosomes is transferred clockwise or counterclockwise. Conjugation, replication, or the rolling cycle models. Transfer is initiated by the endonucleolytic nick at a specific site on the F factor. The 5' end strand transferred through conjugation tube into the recipient cell, coupled to the rolling cycle replication. Detailed work on the conjugation was done by using HFR strain by the HW Hayes. HFR stains H for the Hayes. In this stains, the F factor is integrated near the threonine and leucine loci. Okay. First of all, we need to know what is the rolling cycle. Mm, Say for example, this is the F factor. The F pili the F pili of the F positive donor cell make contact with the F negative recipient cells F negative recipient cells and pull the cell together. Genes on the F factor uh, direct the synthesis of conjugation bridge. One strand of the DNA is cleaved at the origin of replication of the F factor. One strand is cleaved at the origin of replication. Okay, this one strand after cleavage will move from the conjugation tube to the next bacteria. Rolling cycle replication transfer one strand of the F factor into the recipient cell. Now this is transferring from one strand is transferring from one bacteria to the other bacteria. Replication of the F factor occur in both cells. It will replicate in both cells. जहाँ से ये स्ट्रैंड निकू जहाँ से ये स्ट्रैंड निकल के इसमें इस तरफ मूव कर रहा है उसके अगेंस्ट यहाँ पे जहाँ जहाँ से रिमूव वो कराता जा रहा है उसके अगेंस्ट ये यहाँ पे रेप्लिकेट करता जा रहा है और जैसे ही ये यहाँ पर आएगा इसके अगेंस्ट इसकी रेप्लिकेशन होना स्टार्ट हो जाएगी. ट्रांसफर ऑफ द एफ फैक्टर इज कंप्लीटेड yielding two F positive bacteria. So for example, this is the complete diagram. The most famous experiment is the interrupted mating experiment by the L.A. Wollman and the Frankos Jacob in 1957. They crossed the F positive with the F negative cells. By using this method, we can also map the genome of the bacteria. Say, for example, we can also map the F factor of the bacteria. Say, for example, this bacteria is F positive, F factor positive, and this bacteria is F factor negative. It contains some genes with the positivity. Say, for example, uh, it can grow on the selected medium. It contains the streptomycin. It contains the Z plus or and the minus strain. Turn T1 plus and the minus strains, they cross the lag plus and the minus strain, they cross the gall plus and the minus strain. So this bacteria is the positive bacteria and this bacteria is the negative bacteria. Genome while passing from positive to the negative, the genome will pass from positive to the negative up to the 0 minutes. There was no transfer of the genome in the 9 minutes. The first Gene was transferred, and after they checked in after 11 minutes, then 18 minutes, and then checked in 25 minutes. What happened at these intervals? What happened at these intervals? On these interval, when they checked after the 0 minutes, there was no transfer. When they checked after 9 minutes, they found that first gene is transferred after 9 minutes. The HFR HAZ is a gene first HAZ genes was first appeared in the recombination and about 9 minutes after mixing the HFR and F strain of bacteria. They continued the experiment after around 11 minutes. The tone lag after that, after 11 minutes, the tone gene was appeared, and after 18, say for example, lag gene was appeared, and after 25 minutes, gal gene was appeared in the genome of the F negative bacteria. These results indicated that the genes from the HFRH were being transferred to the F cells in the specific temporal order, reflecting the order of the genes on the chromosome. So it means as the gene is the first, Ton S gene is the second, lag positive gene is then third, and the gal positive gene is the fourth on the fourth number. So this determines the order of the gene. This is determining in this order the genes are present. Complete transfer of the genome takes about 90 to 100 minutes. Time interval between the transfer of any two marker is good estimate of the physical distance separating markers on the chromosome. We can also we can also map the distance by using the uninterrupted experiment. Linkage can also be determined by the uninterrupted mating experiment. If mating allowed to proceed for 2 hours, what will be the frequency of donor markers in the combination will determine the distance. In the interrupted mating, minutes are used as the mapping unit for linkage, which represent the time intervals between the two markers. One MD, one map distance will be equal to the length of the segment transferred in 1 minute. Hoping it will be enough for today. We will meet inshallah tomorrow. Good luck.